what do you want?
No, nah.
Good.
Yeah, so most everybody should have basics and whatnot. There we go.
Perfect.
And I might, at the very beginning at least, I might have to just step out for a little bit. I have another kind of like brief call, but then I'll check.
Cool. I thought one other question that I kind of had was, how familiar are people with like using like interactive tools like PyMol?
I don't know. So
Okay, I'll ask and you decide based on.
okay.
I think because this is the first homework we usually do in the class, I'm just curious.
Yeah. And what I'm gonna do is, as we get going, I'm gonna probably post up like the links to the previous courses, so part 1, part 2, so people have stuff to refer to. Hey, Ellie. So people are joining now. Hey.
Huh.
Cool. I wonder, let me message some of the attendees, because they'll probably start joining a little bit as well. Okay. And if anybody joins, I'm going to make, Manu, I'm going to make you co-host so you can do all the things.
Perfect.
There you go. Okay, cool. And we should, there was a couple of people who requested to join. There you go. There's Felix. Hey, Felix. There's Yada. Gabe. Cool. Wonderful. Very exciting. All right. Awesome. There's Mark. Mark's calling in from New York. I'm also on the East Coast this week, so.
Oh, cool.
Maddie's here too. Wonderful. Hey, Mark. Nice to see you. Hey, Maddie. Good seeing you too. So what I'm going to do is I'll give a little intro to Manu, if that's okay. If that's all right, and then you can kind of continue that. But this is kind of like the final course as a part of How to Grow Almost Anything. We set it up as an online course, so it's a little bit more open as well, wherever you are in the world. I figure we could go around with some brief introductions. So your name, where you are in the world, and your experience in protein engineering, I think would be really helpful. But I'm really overjoyed to have Manu here, who has taught this class, I don't know how many times, probably 3 times now, something like that.
So. Yeah.
So she comes to us from MIT and is currently working at Future House, helping build and design an AI scientist. An absolutely brilliant human. Previously started a company in, I believe, some sort of like food tech flavor design with a mutual friend, and has like a deep level of experience and understanding in protein engineering. And so, Manu, do you want to introduce yourself a little bit?
Yeah, I guess I don't know how to count how many years I've been at MIT at this point. I would consider myself technically a 3rd-year PhD student, but according to my advisor, a 5th-year PhD student because I did my master's with him. I primarily work on a protein design. I'm sure like a lot of you have heard about like AlphaFold and like all of these styles of models. A lot of my focus both is sort of on the side of building like AI systems, but I generally like designing specific proteins in the lab, and I like to bridge what's in between. So I really am passionate about teaching this class usually at MIT. So I think we have an explosion of models that can model different aspects of proteins. So my primary thesis is focused on how you can use that to understand the immune system and use that to design proteins. But I'm generally passionate about everything protein engineering. So would love to talk. Like, I think since we have a small enough class, I think it would be like if we during the conversations, like if you guys have proteins you're interested in, like we can talk about how the AI tools could be applicable, because I think that's not always very clear or straightforward. And also this field changes like every day.
Wonderful. Yeah. Well, I would just going on down the list, let's popcorn a little bit. So, Ellie, do you want to introduce yourself, your name, your background, or where you're calling in from, and then your experience in protein design?
Hi, my name is Ellie. I am coming from the Bay Area. I'm a postdoc at the U.S. Department of Agriculture, and my background is plant immune signaling and metabolomics. And I don't have much experience working on protein design, but like something of a curiosity, especially in the small molecule binding world.
Cool.
Um,
I think Felix, I think on my screen you're next.
Hey everyone, Felix Lamb. I'm also based in the Bay Area in San Francisco. I'm a microbiologist, a wet lab person by training. I have absolutely zero experience in protein design. Usually I leave that up to the microbes and under selective pressure. There's something called AI that's apparently really big now. So I want to just see how this is literally my first foray into any AI model into protein design.
I think Gabe. I don't think we can hear you. I think maybe we can move on to Yada, if it's okay.
Hi, I'm Yada. I'm based in San Francisco. My background is squarely in ML, and in both in research and now more in applications, currently at a startup. I don't have any experience in proteins, so but I am interested to learn. So happy to be here.
Cool. I think Madison.
Hi guys, I'm Maddie. I am currently bouncing around because I just graduated from Stanford, so just moved out. But my background's in physics and electrical engineering, more on the applied math side, but I don't have any experience in protein design, but super interested in learning more. I just read some of the seminal papers and that's it. And yeah, I think that's me. Is that all the questions?
That's really cool. I'm actually an electronics engineer turned protein designer as well.
Oh, cool.
So I think Mark.
Hey, what up? So my name's Mark. I'm based in Brooklyn, New York. My background, so I've been software engineers, did startups for a while. Beginning of last year, was helping refugees, climate refugees, and wanted to stop climate change disasters from in the first place. So kind of did this massive pivot into the sciences. And so I've been trying to stay away from my computer, to just kind of get in the weeds of I'm trying to get into more electrical hardware stuff now, but been wanting to do a lot more ML protein work. Been interested in methane oxidizing enzymes, which are just super gnarly to work with. So yeah, that's probably not going to touch that today. But actually, I guess one thing that would be for inspiration is been just trying to make a color picker for different chroma proteins. So kind of basically being okay, how can you just kind of with an RGB picker kind of explore a lot of different colors, and maybe is there a workflow where you can kind of loop in a chemistry tool to be kind of do some analysis to guess what the emission spectrum might be.
Yeah, that would be cool. I think this class had a point where they would do a chromophore proteins that they were studying, but I don't know if this is there this year. Yeah. That's very cool. I guess Ryan, I don't know if he's around, but Gabe, if your sound is working, we can. Oh, Ryan is here. Hi.
Yeah. I'm Ryan. My background is operations in drug discovery and development with a little bit of software. So have a very small familiarity with like AlphaFold and like protein purification processes, but the design side I'm less familiar with.
Cool. I think that's everyone. Gabe, it's your sound.
Yeah, can you guys hear me?
Yes, that's what sound.
Awesome. It's going to make today a lot better. Yeah, I'm Gabe Montoya. I'm also based in San Francisco. I feel like a lot of you guys. I have next to zero experience with any design or protein type stuff. I've done kind of biomanufacturing from the side of 3D bioprinting, but not so much with synthetic biology. So, but just definitely really interested to learn more about it. So, pretty excited.
Awesome. Cool. I think what I'm going to do is, so I think usually when we do this class, I like to do it pretty interactive. I think I don't like doing slides. But if you want, I do have slides if that's much more preferable. I pasted a link in the chat here. That's the class page. And under the, I'm going to start sharing my screen right now. Share me. Oh. Let's see if this works. I think Safari is unhappy with me sharing my screen. Oh, can everyone see my screen? Okay, perfect. I'm glad I didn't have to close everything. So this is the class. This is the class page that I wrote. Kind of to keep track of like a lot of the homework that has changed over the last 3 years. But the most important part of it, this homework, this whole page is like I think in these sections. So this I will go over each one of those models and like what each thing does and how you can relate them to protein design. But just as a housekeeping thing, anything that I'm talking about, we'll have a link here. And if there is an option to test this in Colab like online right away, then there is also a link to do that. I also have like a bunch of tools that I've listed. So any tool that I'm using, I'm talking about also will be here. Similarly for like any databases that I'm referring to, also they're linked up here. And like there's a bunch of tutorials here if you're interested in like tutorials that I've like written and like screenshotted, which apparently now refuses to load, but yeah, loads well. There's a bunch of tutorials that I wrote that hopefully might be useful if you're designing very specific kinds of proteins. So housekeeping stuff, that's where things are if you need looking up. And if you have any additional questions or any additional models, I'm hoping to keep this document as like a living document because in the last 3 years, since I started doing the recitation for this class, that every year like things change and every year like the model that I would use to design something keeps changing. So I think we are hoping to keep this as like set of living documents. So if you have any other tools that, as we talk about the class, if you think about proteins that you're interested in designing and if you can find models and tools, I will add them here as well. Cool. Everyone can access this page, right? Right, cool. Awesome. All right, I have really few set of slides. I'm going to start here, but I'm going to be jumping back and forth between the models and kind of the slides that I have. So I think I'm sure everyone has like seen or heard about protein structure, like, AlphaFold, and there's like a lot of like models come out in my field constantly. And so each one of them does one very specific task. And I think when you talk about protein design, I'd like to think about them as a pipeline where you're kind of like designing a bunch of different tools, or tools you use in a specific order to have a greater chance at having sequences that would work in the lab for you. And I think one of the, so central dogma of life, I'm sure everyone is familiar with this, but I think when I think about proteins and thinking about like what problems people are designing. So from a protein structure prediction, for something like AlphaFold or like a bunch of these models, you can give it a protein sequence and it will predict the 3D structure. And proteins also interact with other proteins. Proteins can form big protein assemblies. So depending upon the protein function that you're trying to design for, sometimes you may have to model protein-protein interactions or think about how they function like as an assembly, or whether or not a sequence that you designed would assemble into your protein. I think there's a bunch of different models for doing this task of like sequence to predicting structure, or taking two sequences and then predicting their complex structure. People, like, you might have heard the word multimers, and that's sort of the predictions. And this field in the last few years, since the big, AlphaFold has come out, has had like a list of different models that sort of do the same thing, but very different features that are going to be important to consider as we do protein design. The one that's most famous is AlphaFold, and OpenFold is an open source implementation of AlphaFold that is it's also faster to run, uses like more efficient memory and stuff. It's a combined effort of a bunch of different groups. And there's models called OmegaFold or ESMFold or EigenFold. It's typical, I think, issue with AlphaFold is that there's oftentimes in the protein design pipeline, you have to run AlphaFold multiple times because you're going to use that to confirm that your sequence will fold into the structure that you want. And we use structure almost as a proxy for whether or not the protein is going to function the way you want it to. But AlphaFold, if you want to fold a bunch of hundreds of things, you have to have like a huge GPU compute cluster to even run inference, because there's a couple of reasons why that is. One of the big issues with that is that AlphaFold relies on something called multiple sequence information. What that is basically, if you have a given protein, you kind of use all of the evolutionary information about that sequence to predict the structure. But that means you're searching through a database of millions and millions of sequences to find that evolutionary related set of sequence set. So that takes time. So it also means that it adds to the inference time of AlphaFold. And there's models like OmegaFold, which don't rely on an MSA. So you kind of get predictions that don't depend upon evolutionary information. Or your models like ESMFold, which are kind of like language models, like we have like BUT and GPT. You train similar style models on protein sequences, treating each amino acids kind of things like tokens. And then you use the embedding space of these kinds of language models to kind of predict structure. And these models that don't rely on, reading MSA search, etc., they're extremely fast. Oftentimes you might have to trade accuracy versus like speed. And these are kind of conversations that will happen based on how big your protein is, or how much compute power access you have, or if it matters to you that you need to get a really, really accurate structure. So a lot of these kinds of decisions influence how these models get utilized in terms of like design. And I think it's nice to have an intuition of like which kind of tool to pick up when you're thinking of designing a specific protein. So one of the cool things that I want to show as a result of like being able to do kind of fast structure predictions is, I don't know if anyone has seen this thing from Meta. It's called the Metagenomic Atlas. I'm going to paste it here if folks don't have access to it. This has, because one, you can fold sequences more efficiently, it has hundreds and hundreds of millions of protein-like structures pre-predicted of sequences that you find in your databases. Like you can scroll and I can see millions of structures. And I think like these, like some of these kinds are only enabled by like, being able to be more efficient with like being able to compute predict structures. But I think, so that's what like protein prediction structures, where you have proteins that you're trying to predict, and you can predict maybe a protein interacting with another protein. So some models will solve this problem. And then you have other things where, proteins can also interact with DNA. Famous example being CRISPR. Or proteins can interact with some small molecules or ligands. There's a bunch of like people who want to design like enzymes that bind to specific ligands, or folks who are designing ligands that can bind to specific proteins, which are like small molecule drugs. So proteins, like some proteins you're designing may not just be protein-protein interactions, or may not be just protein in isolation. Most often it's either interacting with one of these elements. So more recently, we've had models called like all atom prediction models, where you try to predict the structure of the DNA or the proteins all at once. So instead of predicting just the structure of like a protein itself, like you try to predict the structure completely, like all as like a full set of atoms, 3D positions you can predict. And we'll talk about like why this could be useful in design downstream. I think it's very specific to the kind of protein that you're trying to design. And I think another sort of that point to make here is that like, I think the reason I'm doing this is not to sort of like, overwhelm you, but it's kind of like showing you that like, hey, there's a lot of tools that do something similar, or they do very specific tasks, and of designing something involves like a combination of these set of tools. I think these are some of the more famous tools that you might see people using in protein design based papers. For example, as I was talking about it, you can have protein language models. You take big millions and millions and millions of protein sequences, and you can train GPT-2 style models or BERT style models on these kinds of sequence databases, and then you can use that to do a couple of things. You can use the embeddings of this model to predict specific properties of a protein, assuming you have a supervised data set, or you can use that to kind of, understand kind of sequence likelihoods of a specific protein sequence, or like a mutation that you're interested in. And there's recently models which use protein structural elements as tokens and stuff like your amino acids. There's also tools like FoldSeq, where if you have a bunch of protein structures and you're trying to compare them, like FoldSeq is a very fast way of doing this comparison. MPNN is like another thing that you'll see in models where, say you know the protein structure, the opposite problem of AlphaFold, where you know the protein structure, and now you're trying to predict the sequence that would fold into that structure. So there are these models called protein MPNN, and there's also models called ESM inverse fold. So the ESM model I'm talking about is actually this metagenomic atlas that I'm referring to here. So you can use this model to also predict, given a backbone, given a structure of the protein sequence, like, can you predict the sequence that would make it, or can you create more sequences that would also fold into that protein structure? And I will definitely go on how these are used. And then there's models like diffusion style, like things that if you're familiar with stable diffusion, like these kinds of image-based diffusion models. Similarly, people try to do this for protein structures, where you're trying to diffuse an entire structure from scratch, both to create novel structures and also to create drugs and all kinds of other fun things like scaffolding some important sites of your protein. And there's also models for docking. So for those who are not familiar with what docking is, like, say I give you a protein and I give you a small molecule. Docking lets you figure out where the small molecule might interact with the protein. This is important, like both from drugs perspective and also for proteins that they're designing things like enzymes, etc. So there's also these models like DiffDock that do specific tasks like this. And most of protein design is like coming up with intelligent combinations of using them, along with some kind of high throughput assay in the lab. And it depends from protein to protein in which order you're going to use these tools, and if you're going to use like some of the more expensive tools. Like, for example, if your assay is easy to run and you can run 1,000 things in like a week, there is no reason for you to try and model something that's computationally expensive, like fold sequences, etc. So I think it's very dependent upon what the protein family is. I'm just going to take a pause here because I know I speak very fast. So if anyone has questions for a bit before I move on. Yeah.
I kind of have I guess just a reflection, or just to check I understand a few things. So like one thing I kind of remember hearing from folks doing protein design is, like, I think you were saying it kind of at the beginning with pipeline, and also like the speed of different models, and people using things like ESM folds to like generate a lot of sample or like guesses, and then like maybe having some other tool that's like giving you a score, and then from there you're using like AlphaFold later. I guess like, is there a so with AlphaFold, I guess like when you say you wanted to make sure that it's like the correct structure, I guess like how are you determining that? Is it like, are you, I guess like what's an example?
Yeah.
Of how you
so
like,
I think
know it's good?
So for example, right, like say I have, so say I'm trying to make, I don't know, carbonic anhydrase more thermostable. and I know the PDB structure of carbonic anhydrase. Like if I go look in the PDB right now, I might be able to find the structure of carbonic anhydrase. My goal now is to find a sequence that both functions as a carbonic anhydrase, but will function at a higher temperature. And so now I know a ground truth structure because I know the carbonic anhydrase will fold into this specific structure. So what I can do is I can go to a model that sequence recovery models where I can give it just the backbone trace, essentially the geometry of the protein, and then that model can generate a bunch of sequences. So these are all graph neural networks for folks from machine learning, so that you can model all of these proteins like structures as graphs, and then you find a sequence that will fall into that structure. And then what you do is like you then take those sequences and put them through AlphaFold, and AlphaFold is now going to predict a bunch of things, right? Then what you can do is you can align them and see which sequence is close in space to this ground truth sequence that you found already in the database. So that's, again, it's not going to work for every single thing, but for proteins for which you can find the structure, like that's super easy to do. You can look for that. And what another thing that people look for a lot, and we'll go through this as we do like more hands-on stuff, is people also look at how confident AlphaFold is at its prediction, like how confident it thinks its prediction is. And the higher confidence it has, the more empirical likelihood of it might work in the lab. But I think this is a point that makes it very complicated, is that we can get in silico metrics, like computationally I can say these are like XYZ metrics that give me some guarantee of its function, but it's never something that like I can guarantee 100%. So this is why I think most of protein design involves like having some kind of intelligent pipeline along with like assays that you can work on.
So quick follow-up question. Oh, sorry.
Yeah.
Someone else want to jump in?
Yeah, I'm curious about what you said about confidence, because I know there's issues with calibration of confidence scores, all of those things. So I'm curious what type of work has been going on with that.
I think my field uses this score metric a lot, and I think to some protein families, like some kind of proteins have been shown to have success in the lab because the ground truth for everything in biology is in the lab. So I think there's just been empirical evidence for it, like I think is the way I would put it. I think it's also like, right, like if you're 80% confident that this thing is functional, it might be that like 81% is not going to make a much difference to you. So I think it's sort of like on what that delta is as well. And one of the other things to note is that I'm going to like, AlphaFold has like a set, I'm pasting this here as well. They pre-compute a bunch of like sequences and their structures. Again, it's computationally intensive. So there's AlphaFold database that you can look at. And one of the things that, so if I say, in a protein, like maybe I will share my screen. I'll be doing this quite often during the call, so forgive me. So this is like a protein sequence, protein that's like AlphaFold predictions. This is an AlphaFold DB. And if you look at the protein structure, I can scroll in. It's confident only in specific residues. So if you notice that like every single time there's a loopy part of your protein, like it says like it's like low confidence, and if it's like orange, like very low confidence. So sometimes if your protein is extremely like loopy, there's a bunch of things like transcription factors, like things in our body, whose structure is like super, like, I don't know, it's super like messy in the sense of it's mostly made up of loops in some cases, and those it can't do a good job predicting. I'm trying to find an example, but I'll find it. But like those kinds of like things you may not like use AlphaFold for, because if you take your original structure and you like just run it through AlphaFold, and if AlphaFold is not confident in like most regions of the protein, then it's like confidence doesn't matter at that point. And this also kind of brings up an issue of using AlphaFold as an in silico metric, which means that like any parts of your protein that are loopy or flexible, AlphaFold is not going to do a good job of modeling it. So I think in those cases, you don't trust the in silico predictions. So that confidence we get is per residue. So that also allows us to like have some guidance on like where to go. there's a lot of proteins where even if you have a confidence score, even if there's a bunch of proteins I've designed where it's like 90% confident that it's going to fall into that structure, but that doesn't mean anything. Because if you think about how, and if you've ever looked at the proteins, you'll notice that most proteins are made up of like very few set of folds, because you can have millions of sequences, but if you look at the structural like universe of all proteins, there's common elements and like common alphabet within which they're made up of. So a lot of sequences, even if they fall into the same thing, may not do the same thing that you think, like, right? Having a same structure doesn't mean that it does the same thing in biology, right? It may do something completely different, and it's very dependent upon that sequence that's making that structure. So oftentimes, yes, we can use these prediction models like as a good metric for the first set of screens, but it's no guarantee on whether or not it would work. So I think that's one way to answer. I will also talk about like how we sort of not guarantee, but like have some confidence of structure in some of these design-based models of function. So I thought maybe like I can go over like one scenario in which like how you'd use these tools, and then like I would go through one or two scenarios, and maybe then we can switch into like actually using the tools and seeing like what happens with various kinds of proteins. So I'm gonna share my screen again. So I think this is an example. I think most of what's exciting, I think this is a very exciting time for how drugs are being made. There's a lot of companies that are now working on like AI-based drug discovery. And an example of how you can use the tools that we're going to actually use today to make a drug target is, for example, drug lead is, think about the COVID-19 spike protein, and the one that we're all familiar with at this point. Spike protein interacts with a protein called human ACE2 receptor on our cell surface. This protein-protein interaction is sort of what enables like COVID infection of a specific cell. So if you're a drug designer and you're trying to make a drug for COVID, like one thing you could do is stop this infection from happening. So you make another protein that would bind to this spike protein of COVID, and then that would hopefully inhibit its interaction with the human ACE2 receptor and prevent its entry into the cells, for example. So it's like one target, right? And that process is like knowing which protein-protein interaction to inhibit and like finding which protein to target to make a drug for is also not an easy process. But let's assume here that you know which protein you're trying to target and you know where on the protein you want to like make a drug for. So when you're making a drug, you're just trying to make another protein that would bind to the spike protein and just stops its function from happening. So what you can do is there's a model called RF diffusion where you can give it a protein structure and you can ask it to make you a binder, and it does a great job. It uses diffusion, like these kinds of stable diffusion style models, diffusion to generate a protein structure that would nicely fit into the pocket of the spike protein. And now, but the model only gives you what the geometry of that protein structure is. It's not going to tell you what sequence is going to make that. For that, you have to go to the protein MPNN model and then get a bunch of sequences. And then once you have the sequences, you can go fold them with AlphaFold. And once you fold them in AlphaFold, you can see a bunch of like confidence scores, like all of these things. And you can then sub-select a set of sequences that you want to test in the lab. And you go test things in the lab, and hopefully some of those things will bind to your protein. So like, right now we're using like 3 tools to do like this job of like trying to find a binder. So it's like one scenario in which like you can use these tools, and in one way in which you use the in silico metrics, which is that like if I made a backbone that I say is a good binder, the sequence falls into that, then I want to be confident that the sequence is actually going to fall into this backbone that this other model tells me is going to bind to the target that I'm interested in. So that's one way we use like AlphaFold confidence score. And it's usually just one of the things that you use before you go test it in a lab. But that's a very common one with these kinds of AI-based pipelines. Any questions at this point?
Yeah, one quick one on the how are you so are you selecting like the residues, or I guess how are you constraining what that protein looks like that's going to bind? Like how are you picking what you want to do? Because I guess you can kind of be like, well, I'm just going to keep making a bigger and bigger protein, or I guess like, or transcription factors like you were mentioning earlier. Like how do you account for things like that?
Yeah, so I think that's why like finding where to bind and like finding a target, like all of these things are like people do a lot of like other kinds of like modeling and like assays and to find where it is. I think sort of problems for a protein designer would start like, I know the protein I want to target already. But if you looked at the thing I was talking about where a COVID protein interacts with the human ACE2 receptor, we have a structure. So this is
right. Or I guess I mean like when you know the structure, like how do you pick like, oh, I want that section and only that section?
Yeah, so I'm trying to show you by example here. So I will also go over how to use this tool, but this is one way that I look for things that interact. So you can, so PDB is a database of all protein structures that we have available. And this is the structure of the interaction that I was just showing you, the spike protein interacting with the human ACE2 receptor. So what I can show you is like, so let's look at this chain. This blue thing is your spike protein, and this green thing is your ACE2 receptor. So this is how the structure of this interaction as it was crystallized. So if I look for where I want to bind, I can see that like things that are closest here, the interface of where these two things interact are here. So if I want to make a binder, I can have the RF diffusion model like focus on these regions that actually are going to be in contact for the interaction. So that's one way you can find where you want to bind. So you can look for a protein-protein interaction that you think is responsible for like infection or other things in the body, and then you can try to target just those regions. Again, it depends, right? This is for designing something for a viral protein. Sometimes it might be different. Sometimes you're trying to target a specific mutation on a protein, like where you have disease mutant, like something has changed on the protein, if for whatever reason, and that is responsible for disease, and that there's like healthy protein that's like that doesn't have a specific changes in it. Now you want to make a binder that like only targets the one that's been mutated and not the one that's like actually healthy. And those kinds of problems are harder. Like they're not easy to predict in silico, and sometimes impossible to predict in silico. So that's why a lot of the drug companies and like drug pipelines like do all of these things, but you still screen thousands and thousands of candidates in the lab. But the hope is that as you go on, like things, hopefully you can generalize or you can fine tune your models and you can create a lot of these things. And one example. So this is a good question, actually, because I think drug design specifically, all of these models do drug design. So say you made a binder and say my sequence, went to the lab, haha, it works and I'm done. Like, but the problem is you're not finished there. You might make 1,000 sequences and you go to the lab, okay, all these 1,000 sequences work, but you don't finish there, right? Because what happens next is like, okay, these 1,000 things bind to COVID. Great, now I have a drug starting point. But thinking about how it's being applied, like you're going to now put it into the human body, like that's where you want the end goal to be. But when you put it into the human body, you have to guarantee that it's not going to interact with other proteins. Like you have so many proteins in your cell, like you are only looking at one protein in context and like you're designing this, but you have thousands of proteins inside your cell. Like you want to make sure that it doesn't cause those like off target like responses. And so maybe only some subset of these sequences will actually are not going to cause that. And then you kind of will do immunogenicity, right? Like this is a thing that you're creating from like a generative AI-based model. It's not a protein that your body is going to recognize as being human. So if your immune system is not going to be happy, you make the sequence. And sometimes the sequence might bind to the target you're interested in, but it also binds to itself, which means that it's going to start clumping up in the body, and the immune system will also not like this aggregation stuff to happen. So now you have all of these other problems that prop up, even though you just now screen for binding. And so this is why often you might want to have like a set of things, a large set of things that bind and do the same thing. So like, I think even if we had in silico metrics, I think people would still create larger and larger data sets of these things. So you can satisfy all these properties. Like another thing is some pharma companies might also care about patent infringement. Like you want your sequence that's doing the drug function you want to be very different, so you can patent the sequence, which is interesting to do with nowadays with like AI-based models. And so I think diversity also then becomes an important factor in being able to design things that are that big. So usually these libraries are pretty huge, and the binding becomes the first step. And this is why a lot of them fail like somewhere in the drug pipeline, like, they cause like some immune response, or they have off-target effects, and there's all of these things that could happen. So I think the AI tools are great to generate leads, but they're not going to make actual drugs. But it is cool that we can do this now. Like, I think what's cool is that in the past, yes, we could do them in the past too. Like we can do them in like MD or like other things, generate these things that bind here. But now you can do them super fast. It takes like 2 minutes to run this. Like I can run it in 2 minutes, go spend like a few seconds on getting the sequences, and now I start with a library that I'm ready to go. Assuming I have enough time to do AlphaFold and stuff. But like now I'm ready to go with the library almost like immediately. And there is also like people who design antibodies, like things, different kinds of proteins that all stick to other proteins. So I think it's like a really exciting time in the field. And hopefully this means that we can get to drug leads and stuff faster. Cool.
I have a question just kind of maybe about the software. So in RF diffusion, you said that, you can just ask it to do something like create a binder. What are other, I guess, like general functions that some of these softwares have? Is it like create something that cleaves this, or that, does this attachment, or things like that? Is that like just a
Yeah, so RF diffusion can also, like, so all of these models, another protein design. So I think I talked about drugs, but that's only one kind of protein design that you do. It's one of the other kinds of protein design you can do is like, okay, you know that this protein is going to interact with, say, I don't know, some small molecule, like some ligand that it interacts with. Or some small part of the protein that interacts with the thing that you're interested in, right? And the rest of the protein is super big, and you want to make a smaller variant of this thing. Like either it's for delivery, like a bunch of other reasons, you want to make a smaller version that does the same thing, that still binds to the same thing. Oftentimes, a lot of parts of the protein are not just for binding to the one thing, they're doing other things, or like, there's a lot of it is stabilizing, holding that interaction in place. So you can do something called scaffolding, where you take a segment of the protein and you can try to generate like the rest of the protein. Like you can generate of any length the rest of the protein that would still have the same scaffolding thing. And then you can go to AlphaFold and make sure that it's like just as aligned as it should be. So that's one way you could do the design as well. So that's you do them, think of them for enzymes, like you think of whatever you design for. You can think of making like smaller versions of things, so you can put them into like capsids or something for delivery. So there's a lot of things you can do by just scaffolding. Right. And that actually brings up a good point on like how we sort of look for function. Is like, say, take the example of an enzyme, right? I'm trying to carbonic anhydrase, because it's a thing I like. Say I know that the reason it functions is because it interacts with a small molecule. What I can do is go look at the protein and see every single residue that's close to the interactions. Like say like there's 5 residues that are interacting closely with this small molecule. And now I want to make a sequence where I hold all those 5 things constant. So I can go to this MPNN models and make a sequence, and then that holds all those 5 things as where they should be. And the hope is that if it's binding and stuff, like maybe that's a good guarantee for function, is that not all of the protein is responsible for doing the action that it's doing. Maybe only some part is. So if you can correctly guess what those are and hold them constant and make a sequence that satisfies whatever property you're interested in, then that's one way you can in silico, I'm going to put that in quotes, guarantee function, that it would do the same task as the original protein did. This is one way we can, if you look at thermostability of an enzyme, you'll see this a lot. Like people want to engineer an enzyme to be more thermostable. Maybe it needs to function in a bioreactor, like something that needs to work at 100 degrees Celsius, for example. But the one you found in nature works at like 40 or something. Now you want to find a sequence that still does the same thing. So one way you can do that is like hold all the positions constant. Use the MPNN model on, if you have a structure, like go make all of the sequence, like mutations, go see if anything is like going to be thermostable. That's one way you can make a library. But again, it's easy to say that because there's a structure. What happens when you don't have a structure? When you don't have a structure, you can use AlphaFold to predict what the structure could potentially look like. And if you're lucky, your structure is like this like nice little confident predict like structure that doesn't have any loops and stuff. And then you're lucky, and then maybe you can use that as a starting point. Or maybe you will not have the choice at all. You have to go to the lab and first generate some data, and then you can use these kinds of language models to fine tune and like create new set of sequence proposals. So I think it's vastly different upon what the protein is and what the function is. So I think that's why this field has so many papers, and often it feels overwhelming. But it's like important to know that like the field is not solved. There's a lot of important problems that need to be solved in silico for us to be able to do design like in one shots.
I have a quick, unless someone else has a question, quick on like data input, like type format question. So like in the viewer you had, I guess like we've been talking about this kind of like diffusion inpainting kind of world where there's like a scaffold maybe that like you're kind of generating around, or, and so you mentioned like the language-based models like ESM folds, but is the input for those still like a coordinate system? Like when you're saying, like, I guess for all of these models, is it expecting that like, here's the residues, here's the like coordinates, like go, rather than like, here's an amino acid sequence?
Yeah, so I think it's for
sense?
Yeah, for language models is the amino acid sequence alone, unless you can, there's models that do structured tokens, but just like protein language models, generally it's amino acid sequences. For anything like, sequence recovery and stuff, we put the coordinate like information in. So maybe it's like actually a good segue into moving into doing more like hands-on stuff, like so we can get familiar with the data formats. So I thought today we can learn how to fold and then learn how to make a sequence given a backbone and how to hold specific positions constant, and then how to use metrics from AlphaFold. And assuming we have enough time and these things can run fast enough, we can try to do a diffusion style like thing. I think one of the coolest parts about the field is that people generally let you have Colab fold notebooks so you can run them in the cloud. So you can get access to these models in the cloud if you want to try them out. Definitely not something you would want to do on a notebook or like on the cloud one by one if you're trying to generate an actual library. But for most folks, it's like a good way to get introduced to like parameters and things you need to set to do what you want it to do. So I think maybe the good starting point is actually understanding what a PDB file is. Like I'll start using this word a lot. PDB file is a format for representing the protein structure. So the thing I was manipulating is just calling a PDB file. So I'm going to share my screen and I kind of walk through how you find a PDB, what that looks like inside, and how you can use this tool that I use to manipulate it. I have a tutorial for this on the link that I shared. If you would like to download and follow along, like I'm going to put a link on if you want to learn how to use the visualization software. They ask you to buy a license, but there's also like free version, academic version of it. So that's the tool that I use. There's various different kinds of molecular visualization tools. That's the most popular one, and that's the one that I'm most familiar with. So that's the one I will use. I also linked on the class note page, but a bunch of other things you can use as well if you don't want to use PyMol. So I'm going to share my screen one more time, and hopefully this time I'll do it side by side. So the database where you can find all of the protein structures is RCS, is one of the servers, like you can look for it here. So if you're looking to find proteins structures, you can look for it here. And if you notice, there's like 221,000 structures. And this is why we need models like AlphaFold, is because these structures, like, they're very hard and sometimes an entire PhD for someone to crystallize a protein structure. So it means that getting insight into a protein structure is sometimes like extremely expensive and takes a long time. So I think things like AlphaFold accelerate this process because I could do this in minutes now for some family of proteins. So if you click on it, it has all of the 227 structures. So say I'm interested in find a protein. I'm going to think about a protein that I like. Let's think the optogenetic like protein, like the channel rhodopsin, which interacts with light and then opens the channel. So this is a structure. So what I can do is like, you can actually manipulate the structure here. If you click on the structure tab in this site, you can actually interact with the structure in here if you don't want to have PyMol, but I like using PyMol because it allows me to export and stuff easily. So if you go here, you can download the file in multiple different ways. So one way you can download the PDB, the structure is in the PDB format, which is the most common format. So I can download it that way. And what I can do is, if you wanted to open it, So you can go to PDB. You can either open it in a visualization software, but I like looking at it in the text file because it actually just looks like a regular text. So this is what the inside of the PDB file looks like. So you have a lot of metadata about the protein structure. You scroll down, and then you can start seeing a bunch of coordinate. Like these are the coordinate frames, like the actual coordinate points that you're looking at. So this is what the model reads. So this is like an atom, and you get which like what the atom type is, what amino acid, and then like what position number. This is the position number, and it gives you all of these like coordinate things. So this is how the PDB format looks like, and basically that means that if you can come up with a good set of coordinates, you can even write your own PDB file. But if you have the file, what you can do is you can open it in PyMo. I will not get a license. So you can pull the file over, and this is what the protein structure looks like. There's a couple of reasons you'd want to know how to use these kinds of tools. Is that this protein, right? So this protein only has one chain. So by that I mean sometimes proteins can be, you can find a crystal structure that's interacting with something else. Earlier example of spike that I was showing has two proteins, the ACE2 protein and the spike protein. So each one is a chain. So the spike protein is one chain. The ACE2 protein is one chain. That's what I mean by when I say chains. So here there's only one protein, but if you wanted to give different color to different chains, you can go to this thing and say like color by different chain. You can also do a bunch of fun things if you want to create like nice little like secondary structural differentiation. It will show you a bunch of different like alpha helices and like these beta sheets and all of these things. But most typically what I use it for is actually to look here, because this is a small like a molecule that this protein interacts with, and I kind of want to find where specific points are that are close to it, and that's kind of why I would use it. And then generally, a lot of the times I'm using it to kind of understand which residues are important. So say I'm clicking on here and I want to know which protein residue is there. One thing I can do is I can go to the top of the file, click on display on sequence, and it displays the entire sequence. So say I'm clicking on this part of the protein, it will highlight up here what the residue amino acid at that position is. So it's like, G at this position. So it'll also tell you which index that is. So it's 261, 262, it's at 263. So this is why I use PyMol sometimes. Like I want to know where things are. And because we will right now go look at some of these models, and it's sometimes useful, say you're asking a sequence recording model to hold some positions constant. You might want to know what the index of that protein is in that sequence. So for those kinds of tasks, this is nice to have. And even if you're trying to design like a binder with RF diffusion, like knowing which part of the protein you want to interact with is also nice to know. Also, generally, it's fun to look at proteins. But this sort of like tools, you can do a lot of things with it. You can, I think there's like, you can create like animations, you can do a bunch of things. But the common thing I use it for is just to poke around in protein. Or if you wanted to export it as like a PNG file, you can export it as a PNG file. So that's kind of a very quick use of like a visualization tool. And it also allows you to look at, sorry?
So I was just wondering, so the molecule in the middle, that came as part of the download, essentially?
Yeah.
Like this is the protein, this is the molecules.
Yeah.
So would most PDB files come with a molecule like that?
Yeah, depending upon what was crystallized, like sometimes you'll have like small molecules, sometimes you have DNA. If you download the CRISPR's enzyme structure, you'll see the DNA and which parts of the protein interact with the DNA as well. So like if the protein was crystallized in context with something, you'll see it. And it depends upon the protein function, right? Like if you look at channel rhodopsin, I would want to crystallize it with this protein because that's the molecule that is responsible for interaction with light. It plays a role in interaction with light and how it responds to light. So that's like, in this case, there is a ligand. So it's nice to have like these kinds of co-crystals, like where you can see it's crystallized with the thing that you're interested in. So it allows you to find out which residues are important, but that might not always happen. But you can look for it in the PDB.
Okay, cool. Yeah, that seems really helpful. Thanks.
Cool. Yeah, I think this is like a quick way. I will use this as we start like talking about AlphaFold. It's nice to put AlphaFold structures here and stuff, but I'm gonna do this. Okay, cool. I am going to switch to back to things. Start going through notebooks, but so far, any more questions on visualization, like why you would use it? So first thing we can do is I have linked a bunch of notebooks here from various authors who come up with who make these models. Also provide like a notebook that we can all use. So is everyone familiar with Colab in general? This is like a Python notebook for those who are not familiar. Python notebook that you can just run in the cloud without having to install anything. So it allows you to just quickly try out a bunch of different models and see what they do. So we're going to do this with AlphaFold. So we're going to try and fold the protein sequence. So I'm going to place this here. I'm going to explain what each parameter means in this thing. Because when you open it, you'll notice that it has a lot of options that you can manipulate and change. So if I share my screen again. Cool. So when you open the notebook, so this is ColabFold. It's efforts of a professor who actually kind of amalgamates all these tools that you can use really well. There's a version of it called Colab Design, which we will also use. But they have all of these Colab notebooks, which makes it easy for me to like set up like runs that I want to quickly look at a structure of a protein for. So if you open this, for those who are not familiar with like ColabFold, like if you open the show code, you'll see all of the code that's just responsible for rendering this. But for most things we do, we just interact with this part of the protein. So what you do is you have to find a protein sequence. So I think what would be nice is to actually take a protein sequence from the PDB and then put it in here and then compare the AlphaFold prediction structure to the actual PDB structure. So I need to find a small protein structure, and I can't think of one. I think I can zinc finger protein. Cool. So this is a protein that often interacts with the DNA. So zinc finger is also interacts. Zinc is a cofactor of this protein. I'm trying to find a nice one. I will pick this one. So if you want to just quickly see the sequence that made this, you can just do this. Like you can click on display files and click on see sequence. So that's one way you can see the protein sequence. So what I'm doing here is just going here and then clicking on FASTA sequence and then getting the sequence. And you can paste the sequence, and all you have to do is like, so there's something called template. So what template means is, so template would basically be like, sometimes, right, if your sequence is similar or like, it might fall into the same structure of some kind. So templates are kind of finding like a existing scaffold or things that you're looking for, like reducing what you're looking at. But I'm going to leave it alone because the cool part of AlphaFold is not relying on any prior information like this, apart from my mistakes. Another option you'll see is something called relax. For now, we're going to set it to 0. But what relax means actually is, I think, is an important point for proteins is that if you look at this thing that I was looking at earlier, proteins, amino acids, like each amino acid has about 14 atoms, like at the max, like has 14 atoms. Some of those atoms are what we call side chains. So I think I can see them all if I wanted to show you those. Maybe I'm not doing a good job. But like some of these amino acids have like something called side chains. So what we call backbone is earlier when I was looking at this protein structure. So let me also put that back again, because put this back again. Oh, I guess I still had the protein set up. Okay, I'm gonna have to close this and bring it back. So I'll go back and open this. So this thing that you're looking at, this shape, you can just get like 4 atoms and then get this trace of what this shape is going to look like. Sidechain atoms are going to be things that protrude out of these areas, sort of like this. And when I said relax, that these side chains can be in different configurations, and each one can be valid. Like in some sense, like some of the chains bonds can be in one place, in like one conformation, another thing, another side. What relax is doing is trying to find the minimal configuration that you can of that side chain positioning. From physics perspective, and find like a good configuration of those side chains. Obviously, if you think about it, it's a very big combinatorial space of like making sure that side chains can be in a specific orientation and not clash with the other atoms nearby and stuff. So that's kind of what we call relax here. So that's what this is doing, is that because AlphaFold is predicting all the atom positions, like it's trying to relax is sort of a way for you to ensure that you find the lowest configuration of this like sidechain atoms. So for now, I'm going to set it to 0 because relax takes a while.
I think Yada had a question.
Oh,
So-
yeah, sure.
Yeah. Super basic question. If you go back to the visual, I didn't see your cursor when you said where the sidechain was. Could you just confirm where?
Yeah, so if you look at the protein, like protein has a shape. You're looking at the shape of this protein. It's typically each amino acid has a set of, like, so if I look here, the sequence, so this L amino acid, like, will have both the backbone set of what we call backbone atoms and sidechain atoms. Backbone atoms are like atoms that all of the amino acids would have. And if you look at just the backbone atoms, you can see almost the entire structure just by positioning just the backbone atoms. You can get this trace of what this geometry looks like. But sidechain things are kind of, sidechain atoms are very important for a protein's function. And what sidechains do is they're kind of like, I'm trying to find a good sidechain figure.
Yada, I just posted a link where it shows what she means by sidechains. It's two-dimensional structures in this case, but it's just basically whatever isn't like your standard amino acid where it's like NH2, C, COOH. Like that's the main chain, and then the side chain is kind of like, yeah, exactly. This kind of thing.
Got it. Okay. Thanks.
And it's what gives charge or polarity or hydrophobicity, hydrophilicity, those kind of things. It adds additional properties to the amino acids.
Yeah. Yeah, so when I say relax, it's just the side chain. I'm trying to find like a example of like different configurations, but maybe I won't get lucky with this. Okay, so I guess I'm not having any luck finding these various configurations of this as a picture. I'm gonna go back. But for now, basically, when I say relax something, it's just finding good positioning for your side chains. So you can have the same shape, and your amino in your side chains can be in different configurations, essentially. And the side chain configurations determines the energy minima of your thing. I can talk about like physics-based models at the end if you were interested in it. But this has nothing to do with AlphaFold itself. We use like a tool, like physics-based course called Amber, and I can talk about what Amber, Rosetta, and these things mean at the end. And so we set this up, and if you have a sequence that you're interested in folding, we can set it here. We can give it some name. I can say this is some kind of DNA binding protein. You don't have to. You can set it to run. So you can click hit run on the left side if you're not familiar with CollabFold. So once that specific part of the code is run, you'll see a small like green checkbox on the left. Next, what we're going to do is we're going to install all of the libraries that AlphaFold needs. This step usually takes a few minutes if you're doing it. But if you're working on a compute cluster, you can just do it at you don't have to do it every single time. But we're only going to do this in the notebook, so we're going to run this. I'm going to wait 2 minutes. So if anyone has any questions, we can talk about it now.
I have a compute cluster. Yeah, how hard is it to translate a Colab notebook like this and run on a compute cluster? Are these libraries pretty much set up to like handle parallelizing GPUs and whatever else magic needs to happen?
Unfortunately, no, I wish. So what's happened? Yeah, I think so. If you think about like what we are looking at right now, like CollabFold, what it's helping you do is like it's almost making everything into like an API like things. But if you were to set up your own cluster, And you also need to set up your own, this second thing I'm talking about, the MSA, this multiple sequence alignment, which is the evolutionary like information about your protein sequence. That requires you to search millions of sequences. And to actually just have your own MSA server set up, you need a machine with at least like about 1024 gigabytes of RAM as a context. Like you hold all of it in RAM, so you can do fast queries on top of it. Otherwise, it will be an extremely slow process. And I think just to put that in perspective, what this model is doing for you is that it's turning that into an API and they run it on their servers. But if you have to set it up, this is all of this nightmare you have to deal with. But I think if you're like a big, yeah. Well, it depends. If you're going to be someone who uses it a lot, I think sometimes it's worthwhile. Like in my lab, I use it, so I set it up. It's just a lot of like DevOps engineers. So what we are doing here is we're kind of just setting the MSA, like just trying to get the server to like take our protein sequence that we have here and find all of the evolutionarily similar sequences is one way to think about MSA. So it's going to go look at this and this, and then this is like making sure that like you want to unpaired and paired basically means that whether or not you're going to pick them up from the same species or like not, like a bunch of options. You can use the default option for most cases that you're using things for, unless there's something very specific that you want to use AlphaFold for. So I just usually use the default here. And advanced settings. So you see when you click on advanced settings, there's an automatic option, and then you see a bunch of these other ones. So for example, AlphaFold 2 multimer is, say if you want to predict the structure of 2 sequences and not 1 sequence. One thing you can set is like AlphaFold V3 is currently the best one, so you pick that one. So a bunch of these are different like weights of the model that you're trying to run. So I'll set it to auto for now. And so number of recycles is also like, I can talk about this in the context of what AlphaFold is doing in the background as well. I'll get back to it. But leave it, you can leave most of these things in the default thing. And I think, so this is like the relaxed thing that I was talking about. So you kind of set max relax iterations. Again, to default, we set the number of relaxed things to be 0. So we're going to leave it there. Max MSA is also like how many sequences you want to get from your MSA server. A dropout is all the machine learning folks might be familiar with this stuff. And save, this is all like options free to whether or not you're going to save things to your Google Drive. And this is when we get to the bottom of the AlphaFold notebook, it makes a plot for you. That's kind of just setting the resolution of that thing. So we can set this to run. So it ran. So now we ran the prediction. So you can see the amount of weights it's downloading. Like the weights of this network itself is 3.5 gigabytes. I think someone had a question.
Oh yeah, I was thinking with dropout, I kind of know of it for training, right? Is this, but it's like doing this during like the prediction cycle? Is that because it's like as it's going and like figuring out the structure, it's like, oh, let me just like chuck out like amino acids to like throw off things? Or,
yeah.
How does dropout work in this context? Yeah.
So I think this is just I'm guessing this is like related to MSA seeds. I have to confirm this actually. That's a good question. I have no idea. I've never used this setting. I always assumed the dropout was meant in terms of the inference. But that's a really good question.
Yeah, I assume so, because it's like, it seems like you can't really train that on the fly. Like you wouldn't be able to like retrain this on the fly.
Yeah. So I'm looking up what they mean by dropout in this case. I'm guessing it's something either related to the MSA or the number of seeds you're making. Yes, I will find an answer for this and like I'll send it back.
Yeah, no sweat. That's also fine.
Yeah, it's a really good question. I think it also kind of shows you like some of the issues in the field is like a lot of the tools are like not less obvious and like what's happening. So we're gonna do this. We're gonna wait.
When you're running this kind of prediction, is there a way to kind of cache older predictions? Or is it you run fresh every time?
So if it's the same sequence, obviously we can cache it. A lot of the times if I'm doing it at work, like if I'm I will catch the prediction, but most of the time you're trying to rerun it because you have a new sequence. Like if you're using AlphaFold as part of your design pipeline, you're definitely going to refold things, unless you have the same sequence for some reason. Right now, AlphaFold, I think so you can use all of this thing as an API. So when I use it as an API call, I don't have to deal with all of this. But in those cases, if it's the same structure that I'm for some reason computing and comparing against, then I cache it. But often I'm probably using it just to a new sequence each time.
Yeah, and if I were to try to like mutate a particular region, but I have some kind of understanding that other structures are preserved, is there a way to go through and mutate specific regions?
So yes, you're asking a very important questions. Yes, you can go here and then change the protein sequence like places. So you can change this to an L and then rerun the whole thing, but you have to rerun the whole thing to predict the structure.
Damn. Okay.
But yeah, one of the important things you're asking is actually a good point, which is that if you make a single amino acid mutation, can AlphaFold like find the difference? Like it sort of comes back to like if you only have one mutation difference between your sequence and then the protein that you're looking at, depending upon where that mutation is, AlphaFold may not be changing its prediction all that much. So you may not find the thing that you're looking for. So when I'm doing mutations, like I usually do them in the physics-based models where I can, maybe it's worthwhile to talk about them too today, is that if I want to get single resolution, like I'm going to change one amino acid to something else, I usually do them in like molecular dynamics, where I will put like some random orientation of these atoms into the physics-based model. And then I try to relax, like I try to find the local energy minima, which takes a long time to run. But I think that's because of like having to do single resolution. But the mutations is a good point. People do use it for that. Like if you have enough variations and you say you have a 10 different proteins that you say are binders, you can fold them together and then you'll see differences in like a thing that's 10 amino acids different than the other one. And you can use that information to figure out which thing you think is going to bind tightly or which thing you have more confidence that you want to try in the lab.
Cool, thank you.
Yeah. Happy to answer more questions because this is definitely going to take at least another couple of minutes.
So in terms of the graphic that is showing this plot of sequences versus positions,
Yeah.
like what's the point of the interim plot if at the end of the day all we really require is kind of the end structure?
Oh, so I think so this is sort of showing you the sequence coverage. So basically I think you want to know, like, you know, sometimes you may not find, say if you're trying to, like, I don't know, mutate one specific part of the protein, you may not find MSA information for this thing, or I think what the plot is showing you is like what sequence coverage is. Here, the thing that we always end up looking at as a metric for when selecting for some things is always just the final PLDTT score. So remember how I said we look for the confidence score. So the confidence score here is the PLDTT score. So this is the value that we usually treat as the confidence of the prediction. So the higher it is, the higher confidence AlphaFold has that's how the structure is going to look if you were to crystallize the protein. And it's been shown to do that if you take unknown crystal structures and when you run it through AlphaFold, which is not, and it's not in its training set, you can see that the higher PLDT things do correlate with what you really see in a crystal structure. And as you can see in our protein, anywhere there's like a nice little flexible region, AlphaFold is not very confident, which happens for a lot of proteins because most proteins are flexible. Most proteins can be in different orientations. Actually, while we wait for them, one cool way to think about protein structures is like, yes, you will see one version of the proteins in weird. So there's this data set called like PDP Flex. This is not real flexible conformations, but when it works, like if you look at the protein, I don't know if people can see the animations. You can see the proteins wiggle around. Most of the times proteins are like have regions that like they don't always stay in the same shape in the when inside the cell. They will move and they will wiggle around and all of these things. And those do matter when it comes to binding, when it comes to function. And we right now do not have a good ability. Like there's a lot of models that people are working on right now to try and being able to model all of these different conformational flexibilities. So you don't have to do MD, but sometimes you do have to use these physics-based models to do this stuff, like to find out what ways it will move. A good way to think about why that would be useful is, I think this was a protein that I was showing. So this is a protein that interacts with light, right? What it does is it interacts with light, and when it interacts with light, this one of the regions of the protein actually moves after interaction with the light and lets ions through. So that's the thing that causes like electric current. Then we use that to actually study the brain. So if you make a new sequence and you're going to say this is channel rhodopsin, this is going to function just the way it does, you want to make sure that it's going to have flexibility in that region, right? You don't want it to be rigid because it does have to move. So a lot of the times those kinds of flexible things are important for the protein function. Like in this case, it is important for this opsin to work the way it does.
So do you have to do, like, how would you get the structure? Yeah, because I guess if you're designing your scaffold or using the scaffolding of PDB and it's the like crystallized form, but you actually want to like scaffold to it in like this other conformation, are you going into PDB somehow? And like, or how are you figuring out the coordinate system of like that other
Also
structure?
a very nice question, because so take opsins, for example, right? To figure out whether or not a specific sequence is going to work like an opsin, I have to go to the lab and set up an assay that takes me a lot of time to test one sequence for. Like it might take me a while to kind of make sure that I can, measure its function. In that case, right, I want to make sure that whatever sequence I'm taking to the lab has guarantee of function, in which case I will go do this thing in physics and try to model as many things in molecular dynamics as I can, which takes hours and hours and hours and hours of time. But say it's easy for me to go test this in the lab, where I can test like 100 things at once and it doesn't matter. It's not going to be that slow. Then I'm not going to do any of this modeling. I'm just going to take sequences right off as they are and go test them, because why would I spend computational cost? So I think it's like oftentimes when I'm designing them, that's the kind of the balance I'm trying to go for, is like expensive compute or like try a bunch of things. Unfortunately, we are not lucky to have like a nice little assay for every single protein family. That's why the pipeline looks very different for different proteins and which order in which you use these tools.
And when you're saying assay, you mean like you can throw a chemical in and if you get a color or something binds, or you have to wait or whatever, there's just like, okay, you got like the conformational structure, like,
yeah,
correct, yeah.
Yeah, so I think like when I say an assay, it's like trying to see if the protein functions. Like in my case of an opsin, I put the protein, I shine some light on it, and I measure the electric current across it. So that only works for the opsin protein structures. Like if I'm looking at an enzyme that's is carbon capture style thing, so then the assay looks very different, right? You might pass like carbon dioxide in a chamber, or you might dissolve carbon dioxide in water. So you change the assay for that protein. So if I can test a lot of things, then great. But that's very dependent upon how easy it is to set up that functional test. But it's not easy for opsins, because you might sometimes study them in neuronal cultures and expensive. Like it takes a bunch of effort to kind of even get them to express inside a cell. So it's like whole set of those kinds of decision making that happens. And if you're in a small funded lab like ours, maybe I don't have enough money to go do expensive assays. So I want to get my answer in as many few shots as possible.
When you said that the process might be different for different proteins, is this like different protein like functions? Like, are you grouping it by function, or what are you grouping it by?
Yeah. That's kind of what I'm saying. Like if you're trying to make it, if you're trying to make a protein binder, is there an echo? I think.
Yeah, I think you need to mute.
Sorry. So what I'm trying to say was like, if you made a protein that is supposed to bind to COVID, then your function looks very different. You'd run like something like a binding assay. So you want to measure whether or not things bind. And so that functional test looks very different than testing for something that interacts with light. So you can set up like large scale binding assays if you want to, but it may not be as easy to test for function of what happens to this protein upon shining light on it, kind of thing. Is that helpful?
Yeah, that makes sense. I can see how that would be just a tougher experiment to run in the lab.
Yeah. So I think this thing has finished running, so which is great. So yeah, this is the completely predicted structure that AlphaFold predicts. And what's cool is that you can see, usually it puts the folder result here. You'll see a zip file, like if you ran it as I was doing it, you'll see for whatever protein that you folded, like a zip folder that has all of these predictions. So it'll do a bunch of seeds, like it'll try to do the prediction multiple times, essentially. So we can pick any one of them. So this is a protein that I just pasted here. And what was this protein called? 6IET. So this is a protein that we know exists in the crystal structure of the in the crystal structures database. And that's the protein. And I can take the protein that was just predicted by AlphaFold. So the pink one is the one that AlphaFold predicted, and the blue one is what the real crystal structure of the protein is. So say if I'm trying to do the design and I'm going to test whether or not the pink protein will put in the lab or not, one of the things I would do is like I would go to this Pymol interface and I'd go to this align option and I'll click on align to molecule and I'll click on this protein. Why does it not let me click? I'll just do this. So you can also, apparently the protein name is too long. You can do this. So you can say align all to this protein. So you can see that the pink and the blue one almost are like overlap pretty well. So it's very close to what the original structure sort of is. So cool, this folds as it should. Maybe this new sequence or whatever sequence I'm looking for is great, and I'll go test it in the lab. So I think that's one thing that you can use the visualization tools for and stuff. And one way you can use the predictions from AlphaFold is like see how close it is to original.
Yeah, that was just Maddie. Maddie needs to leave a little bit early, but we're going to share the recording around. I think that something that I was wondering is, do you have to run individual proteins for multimeric proteins? When you're predicting the structure of multimeric proteins, you have to do one at a time in order to predict folding structure?
No.
Okay.
So luckily, there's a variant of AlphaFold called AlphaFold Multimer. You can give it both the sequences at once. That it's actually cool because like it allows you to kind of sort of use it for binding predictions. Put all of these things in code. It sort of is helpful for doing like binding based screen. So you can put both the sequences at once, and you put a semicolon between your sequence, and then it'll fold them as two different proteins. In your sequence, if you put a semicolon in that notebook, it'll just think there's two proteins you're trying to fold together. So then it'll run the AlphaFold multimer automatically. That's also what RF diffusion sort of you can use. We'll talk about this other model that can just generate a binder. That's one way you can like see if the binding prediction works for you or not. Is just go fold it in AlphaFold. If you thought AlphaFold single thing takes a long time to fold, AlphaFold multimer scales. The computational cost and how much time you're waiting also scales as the sequence increases by a lot. There's some structures that take 10 hours to fold. So I think it just like depends upon how.
Oh my God. Okay, yeah. So this reminds me of like dial-up internet, when you like wait to get on the internet and you're just like waiting for things, and so you like go make a sandwich and then come back. This is amazing.
Yeah, it takes so long. But I think earlier we were talking about some models that you can use. So OmegaFold is much faster. Sometimes you just want to get a confirmation that the 1 or 0 it folds the way you want it to. You don't want the most perfect conference structure for something. So there's faster models you can use. So there's ESMFold and OmegaFold. So they're also listed in the notebooks there if you want to do that. A lot of the times I'm using OmegaFold, and I'm not always AlphaFolding. Like I'm OmegaFolding things, and it's much faster. So I'll just run through it if it's for design. So these are all decisions like we make, because like if a protein is small, like fine, I'll go to AlphaFold. Maybe it's not going to take me that long. But then if the protein is so big, then like maybe I just want a quick look on how it's going to work. But if I'm trying to predict multimer, I can't do that in OmegaFold. I would have to go to AlphaFold. So it's lots of the decisions on what you're trying to do.
I guess just an extension upon that. Is that based on how well it's known and how much the computational resources are being used by people who are querying structures? Because like AlphaFold is really well known, and is that why it's so slow?
Yeah, so I think what's interesting in my field is sort of like there's what I would consider standards like set by specific labs. So there's one famous lab called the Baker Lab at the University of Washington. So they're the more premier protein design group, and they build a lot of the protein design tools. Typically, they will write tools and they're very good at open sourcing them. They'll give you access to them, which is really nice. So once you have code base available and it's from a paper like that, where they also both run a computational thing and then they'll model and they also show you how it functions in the lab, then you sort of can trust the model that like, hey, this is not just like some ML model that I'm saying and claiming that it works based on, I don't know, some accuracy to whatever test said, that I actually made something with it and tested it in the lab. So a lot of Baker Lab papers do that. And so then there's like some understanding of what this could work for. And then it sort of becomes like a standard and then people use it a lot. But it's also important that in this field, if you go on Twitter at any given point in a day, you'll see a new paper. So I think the field moves constantly, and which both is exciting and also hard to keep up with. So they all, some of them do the same things, but maybe one point accuracy is not going to matter to whatever you're doing. What's cool is that hopefully in the future we'll see more like MD things done with machine learning, which is going to be very cool. Okay. I'm not sure how much time we have. If we have time to do our fusion.
Yeah, I mean, we're booked for probably like 3 hours or so. So this is, I would say that's like class 1 in a nutshell is what we just did around protein design, protein engineering. Yeah. And I feel like folks here are pretty equipped with at least lists of models that you can test out, play with, and kind of simulate proteins.
I see.
So
cool.
Now we can mutate those proteins.
Cool. So I think if folks are like, if we have time, then maybe we could go through our diffusion and just like actually try to make maybe a binder.
Great. Yeah, I actually have one quick question.
Yeah.
I was just gonna say if anybody needs a bio break, feel free. But yeah.
So I mean, it seems like I feel like I hear protein interaction has been hard to model, but it feels like that is. I mean, it's just, from what you've been showing, it's like there's all these ways to do scaffolding and whatnot. Is that no longer the case?
So it's not like you can predict protein-protein interactions, PPIs and stuff, but like thinking about like how many structures you have in the crystal structure database, you have 200,000 structures. How many of them structures actually contain protein-protein interactions, like where that's enough of a data set for a model to generalize for any arbitrary protein sequence that you're making? So I think there's like, we do use them, but again, that's why the reason I'm saying quotes for every single one of them.
Yeah.
Like if I was doing a binder, what I would do is like go fold it in AlphaFold, and then again I would go into a physics-based model and I would spend hours looking at it. Or like, there's like those kinds of things that you do. And if you're, so there's proteins like antibodies, like you want to design a specific kind, because most of the other diffusion proteins we're looking at just makes a protein that has like 60 amino acids or 70 amino acids and stuff. Sometimes people make antibodies as drugs. And in those cases, there's specific antibody-based models. Like there's only few thousand. In antibodies, the amount of crystal structures you'll find is even lower, like 7,000 or so. So, and then there's a lot of like language models that are purely trained on antibody sequences. So there's a lot of these kinds of specific things, and a lot of drug companies that do AI actually do antibody design. One of the reasons for it is also what I was saying is that remember how I was talking about immunogenicity on like if you want to take a new protein, put it in the body. Antibodies, like what you can do is like you can design the part of the protein that interacts with the target, and you can put it into the rest of it into a human scaffold. Like if it makes sense. So there's a lot of these decisions people make on what kind of protein to make, what model to use. If it's an antibody, I'm not probably using these things. On like like what are the like after you make the binding happen, what are the considerations? Yeah.
Are there any specific resources that you go to learn more about protein design and engineering, or certain things you kind of take a look at to make those assessments as well, whether or not you are designing an antibody or designing a particular druggable target or whatever it might be?
I go on Twitter mostly, but I think a lot of the things are also experienced from trying to make a bunch of things is what I put it. It comes also like you pick a protein thing and you use all of these tools, and by the end of it, you know where all these tools are going to fail. And the next time you're designing a new protein, you know what to go for right away and what you're looking for. And it also requires, I think this is the part about ML, like in protein design, that I sort of am interested in for my thesis, is the middle part of this is a protein I'm interested in, and here is all of the models that can do this. But I love specific proteins, and then like understanding that is very, very important. Knowing why this protein functions the way it does. Like, you can also like not only look for things that are in contact with the molecule, what you can also see is like, look at the evolutionary trajectory of this protein. Like if there's specific residues that never changed in your evolutionary trajectory and they're conserved, then they're probably important for function. So I think being able to do all these kinds of like knowing all these things is very important. And I think very much like I like starting from the proteins.
What's an example of the physics-based model you mentioned that you
Yeah,
might use?
so think about like before we had AlphaFold, people still had computational models to like predict protein structure. Actually, I have a slide for this. I don't know if it's going to be visible, but we're going, when I talk about energy here, what I'm talking about Gibbs free energy, essentially. So you're trying to reduce this. Basically, lower energy is a good thing. So I had this like thing open. Maybe I'll find it. I'm very messy with my organization. Okay. So one way to think about it is like this map. So in the beginning, what happens is like you start out with your protein like this, like some sequence of amino acids. You don't know the structure yet, like just the sequence, what we call primary structure. So you just start with a set of sequences. Now, like you want to start folding it like piece by piece. So there's a tool called Rosetta, which computes a lot of things like electrostatics, like it computes things like Lennard-Jones potentials, all of these forces in like energy terms that are dependent upon pairwise distances between your residues. And then you write this energy term as you start with the primary sequence and you kind of start trying conformations. You start to fold some pieces, like, some 3-mers and like some big 9-mers. You start folding it, and each time you measure the energy and you try to go like Monte Carlo style sampling of trying to go in the direction of the energy that's lower. And somewhere along here, you'll see a structure that's like has the lowest possible energy, and that becomes your crystal prediction structure. But you can see that like as the protein sequence is bigger, you keep trying all these like combinatorics of like Monte Carlo search of like trying to find this lower energy conformation, and that takes a long time, which is like nice because I'm complaining about AlphaFold being slow, but it was even much, much slower before. So that's kind of what I mean by energy. Is like when I say energy, we're computing a bunch of these like terms that are pairwise dependent upon, like you look at pairwise distances, and that's what you're looking for.
Yeah, that makes sense.
If you're interested in like the ML side of things, one of the reasons we look for the pairwise stuff is something called contacts, like between proteins. Like anything less than 4 angstroms is like a contact. So if you say pairwise, like at things like people used in the past used to find where those contacts were, and they would go into physics and relax them before AlphaFold. Even AlphaFold's biggest contribution, if you look at AlphaFold 1 before AlphaFold 2, was that people would stop at just predicting which residues contact with which residues, and then they wouldn't try to construct the real crystal structure just from machine learning. They would go into these kinds of models and say, here is a constraint. You can try all the Monte Carlo sampling that you want, like search that you want to do, go ahead. But you can only like make sure that these two things are 5 angstroms or 4 angstroms away from each other. So people would do that. And AlphaFold 1 showed that not only can you just predict the possible distributions over these amino acids, you can also like actually just find the structure directly with like a neural network. That was like a big contribution of AlphaFold 1. AlphaFold 2 is like a whole other like contribution, but I think that's kind of cool because a lot of models used to do that before. And that was the ML computation at that time. I say as if it was a long time ago, it was very recently, but yes, I feel old. But I think the protein structure prediction is like AlphaFold is great, but it's not a solved problem. I think it's important to, if you're interested in ML, like it is not a solved thing. I wouldn't call it solved because conformations, like all of these things still exist,
There's a case for incorporating the physics data into the model. how important is that, I guess, if you're saying it adds lots of time to consider the physics?
Yeah, I think it's like I sort of have been using physics as like an in silico metric or like a filter. If there's a way that you can use that so I can share a paper if you're interested in how you can use physics-based models with these kinds of ASM style stuff. It's kind of like an RL style paper that runs like where you use the physics scores during your design step to kind of guide which way to go, like because like all of these models that are like taking a backbone and sampling something, you can kind of have physics scores. If you can calculate them fast enough, like you can use that to kind of guide your design. That's one way to incorporate it. But most of the times I'm just like making a bunch of things and just going through and like just using the scores all the way at the end. If does it answer your question?
Yes, thank you for the paper.
Yeah. Cool.
I think you kind of alluded to this a little bit before, but you were saying that AlphaFold, if you wanted to do like just a single mutation, probably wouldn't be the right tool for it. So do these tools kind of have like area or certain resolutions that they're good for? Like a physics-based tool would be good for single mutations, and then like is it like 10 mutations AlphaFold would be good for, like general groupings like that?
So yeah, I think again, in my opinion, that also depends upon the structure. There's some papers that also say that like AlphaFold can predict the effect of like single mutations on protein stability. I personally have seen that depending upon what the task is, sometimes I at least do 10 amino acids difference for me to like trust that. And that's very different. Like I'm probably not using them for the same reasons that other protein designers are using them. But most of the time, if I'm making something, I'm looking for like 10 or so things. Like at least some of the proteins I was interested in designing, I kept seeing the same like predictions, even though I was changing things and I was like not happy with it. So for me, it was a threshold of like around 10 when I saw the things. If you're interested in where those kind of limits are, I'm sorry to add papers, but there's a paper. Predictions during effects of mutation. Yeah.
Cool, thank you.
Yeah, cool. Awesome. Yeah, so I figured we can just do RF diffusion and then maybe we can talk about any other. I think it's more fun to talk about proteins designs like itself, but than running a model. But I'm gonna share my screen here. So this is the RF diffusion notebook. If you want to try diffusion here, you could use this notebook to run RF diffusion. And I think so the hardest part about using these notebooks is knowing what all of these terms mean, actually. So what we can do here is, so I'm trying to design like, say, one design of some 100 amino acid sequence. And then I'm going to say, hopefully this works for me. Yeah. So that's like a protein that I'm interested in, the PDB ID. Or whatever protein I'm interested in. And I'm going to say some A50, A6. It's kind of like, remember how I was showing you a protein? And then I was like, oh, this area is the one that's interacting with the spike. So those are hotspots. Like you kind of say, here is where I'm trying to make a binder for, if you want to do that. So I'm picking some random numbers and I can hit enter. So this notebook for me sometimes has failed. I'm really hoping it doesn't. So diffusion is going to run. I'm going to get some output.
With the hotspots, those are going to be included in the structure, or are those like kind of your scaffolding point? It's like, hey, or yeah, I guess this is not scaffolding. This is like expect that amino acid sequence in painting, I guess maybe, or out painting.
This is just diffusing a sample. Like, so if thinking about like how we were making all this like pictures for stable diffusion, it's sort of like you're making 100 ml protein. That is going to start from noise and it's going to make a thing that looks like a protein.
Right. I was wondering what the hotspots, I guess, are. So that's like, you're kind of setting up initial structure to like build off or diffuse from. So like out painting from the hotspots.
Yeah, yes. Basically, the reason you do that is that if you unconditionally sample and you can just say no hotspots, then the diffusion takes a long time, right? Like it's going to try some random region. And you want to make sure that like, if you know the binding pocket, because not all parts of the protein are like nice for receiving like a binding. target. So for those kinds of things, you want to kind of point it at a specific hotspot. So if you look at their paper, this is what they mean by interface hotspot. So you are trying to make a protein that binds here, and you kind of point out what those orange spots are. So the diffusion model only tries to make things there. Because if I'm doing this, like if I'm using auto diffusion to make something, I'm not running it once. I'm running it hundreds and hundreds of times. So each thing takes a few minutes. So because what I want to do is create a bunch of different structures that can fit there, and then make sequences for all those different structures, and then go fold them and filter them. Like a huge computationally intensive process of making a binder. So I don't want to just explore everywhere on the protein structure. So I pick specific regions that could be good pockets to bind it.
My experimental hat is on, and I'm just trying to figure out, your output of this process, how you generate basically like a library of constructs, and then do some sort of direct evolution or something to actually screen those kind of concepts. Like how do you restrict that space, and then like what does the workflow look like for that? Because we're doing all these things like one by one by one by one. Yeah. How do you line that up?
So I think in this case, right, like let's take the binder example in this case. So what I would do is like I'd find a bunch of hotspots, like, you can read about it in their paper. They provide some hotspots for their targets. You make a binder there, and you make a thing that has a beta sheet and 2 alpha helices, 3 alpha. I'll do like say 1,000 versions of this. And then I use another model to find a sequence that folds that way. Great. And then I'll go alpha fold everything. So if you see in the paper, they say alpha tool folding. So now what I'll do is like, I have all these structures that I made with diffusion. I have all these sequences I proposed for this. I have now say 1,000 structures I need to alpha fold with the other binding partner. So I do multimer prediction. Again, very, very expensive. Once I have all the multimer predictions, there's some empirical evidence. So in this paper, they will talk about it. There's a specific AlphaFold conference sort of score that they're looking at. AlphaFold gives them like a predicted, like PAE score, predicted alignment error. So they use that score and say that if that score is below like some number, they have had empirical success in the lab for binding. So I would do all of this. I do AlphaFold multimer. I've spent some days at this point doing compute, and now I have this huge set of sequences along with their AlphaFold scores. And then I say my whatever binding affinity assay I have in the lab, I can take a set of like 1,000 things I can go order. But as you can see, like this takes to do this for one protein takes forever.
Yeah. It's just the time required and the compute required to get there. It's kind of wild. Yeah.
Yeah, I think this is something that I think is like a barrier to people using them. If you're not a computational lab, and if you're not a wet lab and a computational lab at the same time, there is a non-significant amount of effort that needs to go in to get these things set up just to run inference.
How do you bring in experimental data back into your design process? Because it's like, okay, you've generated all these structures. I wonder if there's like a fine tuning step you can almost do to be like, okay, these definitely didn't work. Let me do is that a possibility?
I have not seen people do that on like structure-based models a lot. What people do is like you made 1,000 sequences, and you get some data on the 1,000 sequences. You go to these like ESM style like language models, and you get the embeddings from like a language model token. Like you embed the sequence, and then you try to do supervised prediction that way. And that's one way. So you can do those kinds of things. I've not really seen people like go back and somehow influence the decision making at this step of structure, because like at least in this case, the sequence is coming from another model entirely. So people try to do fine tuning of things, but also like thinking about like these are not simple small models to like fine tune over.
I think but
yeah.
To your ESM point though, so it's like you have these failures. It's like you have the amino acid sequences. So you go back to ESM fold and you encode those. So now you have like these embeddings. So then it's to say like, if you generate a bunch of new ones, are you always like running those structures kind of through embeddings and being like, okay, if it's similar, like do a similarity distance check and being like, okay, whatever's close to an embedding, like that's a miss. Like, is that how you think about it? When you're thinking about like kind of embedding path?
One thing you can do is like if you screen 1,000 things, you can train a classifier. And then now,
Mhm.
like you train a supervised classifier on top of the embeddings, and then you can use that, or you can find the whole thing. Yeah.
That's wild.
Yeah,
That's
there's
cool.
a lot. Yeah, there's variations of application of these embeddings. If you're interested, like I'm gonna put this GitHub here. This is one of those ones, like they've used this for almost everything. Oh, that's that paper. Cool. This diffusion. Aha, it failed. It made a stupid thing. Huh. That's where it thinks.
So,
That's not a valid thing.
so like you've seen enough structures by now where you're like, this isn't gonna work.
Yeah.
So how do you make that kind of inference though? Like, do you, like the next step here, let's say assume it did work. Would you then overlay that on where it's going to interface or where it's going to find?
No, so actually one of the important points is like, so this set of, so again, I think the part of using these models is sort of also like knowing what parameter to set. So you can use RF diffusion to do multiple things. So this is a GitHub. You can do all of these tasks with RF diffusion. You can do motive scaffolding, you can hold things constant, like, or you can do partial diffusion, like the in-paint things, or you can do binder design. Binder design is one of the things. So like, in this case, what I'm doing is like setting, I'm not overlaying it. I can directly predict the structure together. I'm not doing it because it usually takes a long time. So I'm just showing what the diffusion sampling looks like. But you can use this model to say, I want to make a binder at, B100 thing is the chain B of an atom that I'm interested in, of a protein I'm interested in making a binder to. I want you to make me 100 length amino acid sequence that will bind there, and it'll do it together. It'll do it in the pocket. So you don't have to like go put it together later. So it'll just generate it, and you'll get an example that has both the parts in it.
That's really cool.
Yeah, it's like this model. I think actually there's a bunch of examples if you're interested. It's cool how many things that this diffusion model can do, which I think is awesome. I don't know if there's an example here, but yeah, there's like examples on how to run all of them. But like it's like the way of like these flags are set is like it's whole thing. But you typically like you run diffusion, but it's also an important point. Like I'm looking at it and I can tell you that that's not going to look like a natural protein. But maybe when I'm running inference, I'm not looking at every single 1,000 things by hand. And that's why I'm making like thousands of sequences and stuff, and then hoping that the AlphaFold captures it at the filtering step. Typically, if I'm doing this and I see this, I would never actually look at it with my eyes till at the end step. Because I'm just going to finish all the steps and then come to it. Like, as I said, like they use MPNN, like here they only have like coordinates of like the backbones, like essentially just the pure geometry, not the actual sequence that is making it. Like, because if I download this right now from their model, I think it's this one. And if you open it and see what sequences were inside it, you'll notice that it all puts like glycines here. It's just GGGGG. So it's not the actual sequence. So to get the actual sequence, you run another model. It's called MPNN. You run another model. And in MPNN, you can do things like, okay, I don't want you to put like cysteines, like you can do that. Or I want you to like, be as close to the original distribution. So it's the temperature really low. I want you to make me 8 sequences that all fold into the same structure. So you use another model to get the sequences. And then now that you have a sequence, you go then do AlphaFold to filter. So computationally super expensive.
Just to make sure I follow that, you just said that RF diffusion only will like generate the GGG like every time. You always have to use protein MPNN after.
Yeah. So you do protein
MPNN. Cool. That's pretty important to know. Okay.
Yeah.
That's pretty funny.
Yeah, you do the MPNN and then you take the sequence MPNN gives you. So you look at the sequences it's giving you. Again, this is a stupid sequence. In theory, too many alanines, but like you'll take it and then you'll put it into AlphaFold, and then you get like this like prediction together with the target.
So weird. I'm surprised that that's not like a combined step or something.
There is new models that do them, like not really. I'm gonna put it in the. If you're interested in doing them together, this is one way to do them. But whether or not you can use, I think like. There's models that are trying to generate them together, sequence and structure together, but like they're not like as easy to use as like RF diffusion with all of these different things. So I think
Mhm.
these are cool like ML like approaches. Hopefully in the future we can just go and make it one shot and not have to do this.
Got it. So that's why you kind of have to do this AlphaFold step after. Because I was like, it seemed like wild that there's so many like sequences, and it's like, oh yeah, well, our fusion doesn't give you amino acids. It's
Yeah.
like, oh, you got to use that protein man. Oh, well, that's just like here's a bunch of guesses. All right, let's go it out.
Yeah, it's like all of these are like, and there's also this other model that I really love. It's called Chroma. It's from this other company called like Generate Bio. So this is like a company that has a model. So if you're an academic, I think you can use it. Otherwise, it's not available for. You have to apply for a license for other things. It's also a diffusion model, and they can do cool things as well. The same idea essentially. You can use it to do like some kinds of designs and things. So this is also really nicely set up. So like you can also add like some kind of classifiers for your generation step and stuff. Super cool. So you can use this one as well. It is also another diffusion model. I mean, all of these are linked actually in this notebook. That this is why I keep adding things to it, because I think this field changes every other week. But
That's fantastic. That's probably like one of the most up-to-date resources then, is you scouring Twitter and like scraping different like GitHub pages.
well, I think we used to, like now I'm just only adding things for which there's a collab notebook. Otherwise, like, yes, paper exists. If we can't use it, what's the point? Yeah, I think so. I sometimes I keep it up to date. I think I update it every year. That's where it's like does it every time for the class. But, yeah, so there's like I think Chroma, you can use them. AlphaFold.
You had mentioned in kind of like the protein MPNN to limit certain amino acids, or can you do different kind of environmental adaptations to proteins as well? Can you kind of constrain the protein space on the basis of it needs to have like pH responsiveness or temperature responsiveness, or are there ways to go about doing that? Because there are certain amino acid structure motifs that would probably be, more favorable in that case.
I think I wish that was possible. I think the problem is like you could, if you can have a data set that you can predict that and you can fine tune the model. But if you have to remember that protein MPNN is only trained on like tens of thousands of protein structures to predict sequence. So there's like all of these issues, like, there's also another model called ESM, again, the same ESM, inverse fold, that it trains the model on predicting like sequence given a structure from all of the millions of alpha folded structures. Now, you're kind of, I think whether or not that actually translate to expression or things in the lab is no, who knows?
Got it. Yeah, got it. But I guess you can constrain some of the like amino acids used, or at least like what you were showing is that it picked like a bunch of G's, particularly just because then it was straight and then it would like fit in that region. Is that the case?
Actually, like, can you repeat that question? I think I missed.
Well, it was just like a series of glycine. So I guess I'm just trying to figure out like why in terms of the diffusion model was it just chaining together a bunch of glycine for that particular region that you were trying to specify?
So everything out of viral diffusion probably comes out as a glycine. Like, I think so. This is like a thing, right? If you're representing all 14 atoms, like you think about embeddings of like the size of representation in your model increases, like as you consider more and more. So a lot of the times people have wrote these models, they've done them on like backbones and stuff. Like people are getting there. People are trying to do all atom things, like, trying to beat this, like, but they're expensive. They're like going to be huge parameter models. But people have done a lot of this in the backbones, that more and more now we're going to see everyone predicting things all at once. I don't know if everyone has seen the AlphaFold 3 Twitter things, but AlphaFold 3 came out, which does like predicts like protein structure along with some kind of a ligand or like a DNA and stuff, but they didn't really give you access to their GitHub. Instead, they just give you a URL where you can have very limited ability to do things. Since I think you guys will appreciate it now, because remember I was saying that like I will have to fold hundreds of things sometimes to screen if things are going to work or not. AlphaFold 3 access that they gave, they only allowed you to make 10 calls or so and stuff like in a day. So a lot of things, but they said they would open source it. We'll see if that happens.
I've been following along.
Yeah.
There's some people that are like actively trying to recreate it open source just from the paper. It's been pretty fun to watch. I'm just like in the Discord, like have no idea really what's happening, but trying to follow along. Lucid Rains, I'll drop it in the
yeah, I saw Lucid Rains like right. Yeah, it's like it's cool. What's cool about the community is like there's a lot of folks who care about open sourcing them, because I think it's not just pharma companies that need this. Like academic labs could also use them for whatever things that they're studying, and we need easy to use access to these kinds of models and stuff. Cool. I think those are the main models that I wanted to talk about. If you have other like specific proteins we want to discuss, happy to. If not, there's also other long set of models in that notebook that I'm happy to go through.
I had a question.
Yeah.
Last time I looked at this, AlphaFold had issues with antibodies.
Yeah, I think the antibody CDR region is like full of loopy areas and stuff. Like there's AlphaFold doesn't do well on some things. Like you don't have a lot of MSAs or like things that are like particularly disordered, like or things that are computationally very flexible. Like all of these things are like the AlphaFold doesn't do a great job. But there's actually a bunch of papers that do CDR loop modeling to do explicitly this. So if you're interested, I can send you a couple of papers on that particular problem. So I will send you like a.
Sorry, I'm thinking.
No worries.
I need like a emoji for thinking. The data set is too small. Like you're talking about only 10,000 proteins for the fusion model.
No, I was talking that about in the context of antibody complexes that you can see in the crystal structures. Like it's like what 7K. I don't know what the current number is, but out of the 200K, I don't think a lot of them are like antibody structures, right?
Exactly. Okay, thank you.
Yeah. Cool. If anyone is interested in like Rosetta, I have the tutorials on that link as well if you want to go through like physics scoring. So if you're interested.
Yeah, that'd be cool. Yeah, I'm also curious if you have any ideas on how, I guess one curiosity I had was with the chromoprotein stuff. I mean, I guess with fluorescent proteins, it's more obvious because there's like spectrum data about it. But like, is there any quick thoughts you have on how to understand what substructure is like responsible for like what wavelength gets emitted and the color that comes off of it? Because I've been wondering if I could use some like diffusion model to kind of like, or like ESM fold, like here's the embeddings. Can you kind of like guess in the middle, like how you get a color because we know these two colors? Like, I don't think that will work, but it's just like the workflow I wanted to make for the sake of learning.
I think if there's like, I'm not, it depends upon what you're starting to start from. Like if there's a chromophore structure that you have, then I think you can just look at the structure and see what things you want to preserve and then try to do MPLN sampling. Also, hopefully it's like easy enough to test them at scale, because I think anything like with that level, like I think anything like with some complicated like chemistry is like always hard to do. There's like some enzymes where conformation matters, binding to something else matters, and like, if you have a structure, then like happy to like look at it actually.
The thinking emoji.
I am curious, I guess, at a high level, how you would do like the Rosetta.
Yeah.
Or if there's like a collab for that.
I made collab notebooks if you want to. Actually, what's cool is like Rosetta has Jupyter notebooks, I think, which you can open in collab nowadays. notebooks. I can put some resources if you're interested in, like, I'm putting all of the PyRosetta notebooks in here. So if you want to learn Rosetta, that's one way. I have a simple set of those things on the site as well, under tutorials. These are all of the Python notebooks that you can use if you want to learn how Rosetta things work. I still use Rosetta for some things, but no longer default things. Yeah, I think it's like really nice is that when I first joined my PhD, I was still using Rosetta default things. It's really cool.
I feel like you can do so much more, so much faster now. But
yeah, it's crazy.
Yeah, I'm wondering. So, like collectively, there was like a homeworld grant for various like protein folding and structures. Is that kind of what you're talking about in terms of the protein structures that you're looking at right now? And running computational pipelines on, like, yours was on carbonic anhydrase, right?
Yeah, but I think I'm trying to do tool building. I wanted to solve this problem that I'm talking about is like everyone designing these proteins seems to be from labs that are not traditionally computational. So I was trying to build tools that people can use without needing to set up GPU and friends stuff for them, like sampling and stuff. Because I'm trying to make it easy for people to access it. So far, that has turned into me collaborating individually with labs, but we'll see what happens there.
Well, thank you for teaching, because you're just kind of like opening up the world of possibility to everybody, and I'm sure each of us is going to go tell other people as well.
Yeah, I think it's like these are like pretty much all the words and things you need to know, and hopefully you have to read the papers if you join Twitter. It's a pretty active community on Twitter. I think I'm happy to share a list of people I follow if you're interested.
Can I post your Twitter or something?
Yeah,
I don't post on Twitter.
Huh? You don't post?
I don't post. I have one follower. I think 2 followers, 1 follower.
I just started. I followed you.
Thank you. I don't even know which account that one is, but thank you. But I don't tweet at all. But I like to, because a lot of I think what's cool is people post summaries. So sometimes I don't have to read the whole paper to kind of just figure out what each paper is doing. Because it's really hard to keep track of what's a groundbreaking thing that changed versus like what's a small insight that I can use during design. Yeah. There's a bunch of conferences that my field sort of has at some of these big ML conferences. Those are also nice to track papers there to kind of see where the field is going.
Super.
Yeah, I guess I've been kind of thinking about this in the context of like, how do you break cell walls, or how do you break through cell wall implants? And like, I don't know if there's anyone who has thoughts on this, but I was trying to think like, okay, why is agrobacterium really good? And like, what is the mechanics of those enzymes? And like, can you make a more effective one, or can you make one that doesn't like trigger immune response in the plant? But it's like, now we're getting into like crazy the multiple dynamics and like mechanisms at play, and I'm like, oh god. I mean, it seems exciting, but yeah, I don't even know how to start to approach that. Like I was like, what is the crowbar for like the cell wall that you can like just pop open? Like is there actually a synthetic alternative for like, I guess in silico generation?
I don't know enough about plants. I don't know enough about plants, but one of the How to Grow projects this year discussion was making lysis proteins that like to open the cell walls for like bacteria. So I think those kinds of I think the mechanism is probably different based on it. I think what you're making is an important point, and what I like to do is sort of like, there's a lot of these complicated molecular interactions that might happen to lead to the protein functioning the way it does. Sometimes you might want to model all of them to know where what your function is going to be. And the way I tend to look at it is I get pieces of the picture the more and more I model things, and you might have some data for some things, more data for other interactions. If you piece the puzzle back together, then you know constraints you can use for all of this. I think all of these models are just like tools, and then you can come up with creative ways to use them, and some papers come up with really creative ways of using them. So
yeah, I'm more appreciating the tool part as well, because it's like, how do you just with that microscopic view, especially if you're resource constrained, how do you just know if you've effectively disintegrated a cell wall and you're testing 1,000 enzymes?
Yeah, there's also this whole I'm not gonna overrule it, but like this idea of like membrane proteins and stuff, like the specific kind of like things you need for them to. There was a new paper as of yesterday that does a good job of like showing how these models do on these kinds of like with these kinds of problems. I'm trying to find it. Yeah, I'm putting it here. If you, I think just to answer that question of like complicated stuff, but this is one other paper that's actually using them to design like very specific family of proteins.
This is fantastic.
Yeah.
We're past my bedtime now, I think too. I know Mark's calling in from the East Coast as well, but this has been absolutely phenomenal, Manu. Seriously, round of applause to you.
Thank you
so much. This
Thank
was
you.
an outstanding overview of protein design and engineering and all the workflows necessary. And I feel like the people here are really well equipped. I am going to stop the video recording now. All right. Thank you so much.